there
But
it
that
is.
means that means that you'll get the audio clip. So you know how to upload to YouTube, right?
Yeah.
Okay, yeah. So you can do that. No problem. Okay. All right. Hey guys. Welcome to the Bio 285 review session. This is the second review session of the week, and it's going to be covering content from week 1. Just the other half of what we didn't cover on Wednesday. And yeah, let's get started. Saeed, you can click the...
Oh, yeah.
I don't see anything changing. Oh. Okay. Is it on full screen? I feel like the slide was taking a minute to load.
Is it? I don't
Yeah.
know how to change screens.
I can't see like the left and right corners. It's like a square, like black squares.
Here, I'll stop sharing. Here, let me pause.
Okay, so I think this is a new recording. So welcome guys, this is the Friday review session for Bio 285. We're covering content from week 1. And let's get started. So this is just to review content that we covered on Wednesday, but I figured it's good to go back and look it over again. So amino acids, typically speaking, they have these few groups that are attached to them. The backbone atoms are composed of the amino group, which is the NH2 group, the central carbon atom, the hydrogen atom, and the carboxyl group. And then the R group, which Laura describes in class, is a variable group. So it varies depending on each amino acid, and it's what gives amino acids their unique properties. And I have just a few questions for you guys, just to see if you remember from last review or from class. But what bonds hold amino acids together? And if anyone wants to unmute or put it in the chat, what bonds they think holds amino acids together? Like what's the name of the bonds? Anyone?
There we go. We got one person saying, I don't know how to pronounce it. Shaida? Is that how you pronounce it? She says peptide bonds.
Perfect. That's the correct answer. So that's correct. So peptide bonds is what holds amino acids together, like each individual amino acid is connected to each other through those bonds. Now, do you remember whether those are covalent or non-covalent bonds? Anyone want to unmute or put it in the chat? I'll just check the chat. Anyone remember what kind of bonds they are, whether they're covalent or non-covalent?
Let's see. Yeah, Shaida said again, covalent.
Perfect. Yes, so they're actually considered to be covalent bonds, and covalent bonds are very strong. And I guess I'll just answer the last one, that during peptide bond formation, which is a dehydration reaction, the byproduct that is released is water. So yeah, that is the spiel on amino acid structure and how peptide bonds are formed. Does anybody have any questions on this so far? Okay, I'll take that as no, and I'll continue. Oh. Okay, and we also looked at last time levels of protein structure. You can see here that we have primary structure, which is just your simple string of beads or the order of amino acids, and they come in different orders depending on the proteins. Hold on just a sec. Hold on just a sec. Okay, and like I was saying, in primary structure, you have the sequence of amino acids listed here, and then secondary structure. Eventually you have enough beads or enough amino acids all lined up in a sequence that they form these structures such as alpha helices and beta sheets. So that's your secondary structure. And then when you have hundreds and hundreds of alpha helices and beta sheets, they will fold together in a unique way to give you tertiary structure. And that forms your polypeptide chain. And then because tertiary structure represents a single polypeptide chain, when we have multiple polypeptide chains linked together, that is what we call quaternary structure. And I don't know where my pointer went, but if you can see with the mouse that this is quaternary structure. And when you have two or more polypeptide chains linked together, that is considered to be your subunits that are within the quaternary structure. And yeah, that's just the overview on levels of structure. Does anybody have any questions on that? I'm going to take that as no, and I'm going to continue. Saeed, there's nothing in the chat. Saeed?
Yeah, there's nothing in the chat. Sorry.
Okay, cool. That's okay. So yeah, I included some diagrams in here. I just am a very visual person. I like to see how things work. And last review, I just noticed people were getting a little tripped up on differences between tertiary and quaternary. But as you can see, it's a kind of the levels of protein structure is a hierarchical, I can't say the word. It's like a hierarchy. So you have your primary structure, sort of if you zoom all the way in down to like the deepest level of detail, you have your primary structure, which is your sequence of amino acids. And then with enough amino acids all linked together, like maybe thousands and thousands, you'll get alpha helices and beta sheets. Then enough of the alpha helices and beta sheets linked together, you get this one giant polypeptide chain, which folds in its unique way. And then you have two or more polypeptide chains, you get the quaternary structure. So it's just like I explained before. I just am a very visual person. I like to include lots of diagrams in here. So you guys are always welcome to go back and look at these. But yeah, it's pretty much the same idea here, shown in pictures. Okay, and so next thing we talked about was domains and subunits. A lot of people get these two terms confused, but they are very different things. So a domain is considered to be a section of protein that folds independently from the rest, and it's a region within one polypeptide chain. And domains usually have a specific function or role. So an example I put was an ATP binding domain on a kinase. And you might not know what that means right now, but once you finish being in this class, you might understand what a kinase does and what its function is. But I just included this example just to think of something in real life. But an ATP binding domain kind of sounds like it has a job or function regarding ATP. And so kinases are special enzymes, which are proteins that facilitate some sort of like catabolic reaction. And so kinases are able to phosphorylate things because they cleave phosphate groups off of ATP. So that's why there would be a domain on the kinase, which is the protein for ATP to bind to. So that's just an example of what a domain is. And then subunits are different because subunits are found in quaternary structure and they're a result of two or more polypeptide chains bonding together. And that's why we call each of those separate sections of the chain a subunit of quaternary structure. And so, yeah, that's the main difference between the two. Does anyone have any questions on that? Okay. And yeah, I just included more examples of what domains are, and so that you guys can get the difference between domains and subunits. But like I said, domains often have a specific function and they fold independently from the protein. And yeah, that's all the slide shows. And as you can see in the bottom, there's like several different domains within that giant protein, SRC. Not too sure what SRC does, but I just thought it was a good example. And I guess a distinction to make in this photo is that like this could probably be like a mega protein composed of like individual smaller proteins, but with zoomed into these smaller proteins, or polypeptide chain is a subunit. Sorry, not a subunit, my bad, is a domain. So yeah, that's just this photo. And Saeed, I don't know if you want to add anything, but that's just all I have to say really about domains and subunits.
No, I think that's pretty much it.
Okay. Does anyone have questions so far? Okay. And yeah, this is just like more information for you guys to read about domains and like how they're different from subunits. And just the main thing to remember is that domains serve different functions, and domains are usually found in like within a single polypeptide chain. And that single polypeptide chain is within a protein's tertiary structure. Okay. Now we're going to transition to talk about the different bonding and interactions based on the different levels of structure. So before we do that, I just thought I would define difference between polar versus nonpolar, or just give a refresher. So something that is polar means that it has an uneven distribution of electrons. And I think in some cases it means asymmetrical, if I remember correctly. Yeah, I think so. And so you can see that down in the example, we have water, and water is considered to be polar because there's an uneven distribution of electrons, since oxygen is highly electronegative, and highly electronegative atoms are greedy and they want to take all the electrons. So you see that the oxygen has a partial negative charge on there. And then that's different from nonpolar because electrons are evenly distributed among the atoms, and the molecules usually are symmetrical, and there's a like charge or the same charge across the molecule. So you see in the example, you have a symmetrical molecule, and it seems that all the electrons are happy and the atoms are sharing them equally amongst each other. And yeah, that's just the main difference between polar versus nonpolar before we get into the bonds and stuff. And oh, did anyone have any questions on what I explained so far? No? Okay. So this is the amino acid chart. And usually for most every exam, she gives you a copy of this, or you guys will probably have your notes because I think the exam's an open note. So you'll be able to look at this. But it just is useful to have because it gives you all the categories of amino acids, and it tells you which ones are nonpolar, polar, and then which ones are positively charged versus positively polar positively charged, excuse me, and then the polar negatively charged, so that you're able to know like how to compare different amino acids based on their size and chemistry. And I thought I would define the hydrophobic effect, which we'll get to when we talk about tertiary structure and just the general overall, which is the general overall shape of a protein. And hydrophobic effect occurs when, while a protein is folding, you'll have your nonpolar groups that clump together and aggregate into the center of the protein, while the polar amino acid groups will be present on the exterior of the protein or on the surface. And I wanted to add that polar molecules are called hydrophilic, whereas nonpolar molecules are called hydrophobic. And that's because like hydrophobic literally means water fearing, and that makes sense for the nonpolar molecules because nonpolar atoms don't really like to associate with polar atoms. If you've ever heard the expression like dissolves like, or things that are similar like to interact with each other. So that's why the nonpolar molecules are called hydrophobic, because and they don't associate with water. Yeah. And I think hydrophilic means water loving. So that's why the polar molecules, they will interact with water or aqueous environments. Yeah. So does anyone have any questions on this slide?
No.
Okay. Thanks. I appreciate the response. It's good. Lets me know there's no questions. Okay. Well, okay. And now I'm going to get into like what the specific interactions are between each of the different levels of structure. So primary structure, like we just said, is a sequence of amino acids, and they're held together by peptide bonds. Peptide bonds are covalent, and yeah, peptide bonds are formed by dehydration synthesis reaction. And yeah, so the main bonds for primary structure, or I'm sorry, the only bonds for primary structure are peptide bonds. Then for secondary structure, the only bonds we care about in there are hydrogen bonds, which happen between backbone atoms. And secondary structure, just like we said, is when you have the formation of alpha helices and beta sheets. And how they're held tightly together is by hydrogen bonding between the amino groups and the carboxylic acid groups, or just the backbone atoms in general. You can just say that. And it's important to know that hydrogen bonds are considered non-covalent bonds. So the non-covalent bonds for this class are weaker than covalent bonds. And a good thing to point out is that the secondary structure does not involve the R group interactions, only amino and carboxyl groups, which are considered to be part of the backbone are interacting. So you don't have any R group interactions there in the secondary structure, only interactions between the backbone. And you can see in the diagram below what the alpha helices and the beta sheets, how they're only the backbones are touching each other. Like none of the R groups are connecting with each other, interacting with each other, at least not in the secondary structure stage here. And yeah, it's the same thing with peptide bonds. They don't interact with the R groups, they're left alone, and it's just between the backbone atoms. Yeah, does anyone have any questions on this?
No.
Was there a question? I just couldn't hear.
I'm all set. Thanks, though.
Okay, cool. All right, Saeed, you're good to go with the next few slides.
Yeah, sure. So I'll start off with R group interactions in the tertiary and quaternary structure. There's pretty much three main categories in this. Your first one's going to be for your polar charged R groups, right? So those are going to make one of two bonds. Either if they're going to be two polar charged amino acids together, they're going to make an ionic bond. So example here we have lysine or aspartic acid or aspartate. Or the other type of bond they can make is an electrostatic interaction. And that's going to be, the difference there is just that's going to be a polar uncharged and a polar charged group. So a lysine and a serine. And that's a point of confusion I remember a lot of people had last semester was the difference between an ionic and an electrostatic bond. So again, ionic is just two charged R groups, and then electrostatic is one charged and then one uncharged. And then your second category is going to be your involving your polar uncharged R groups, and these are just going to be making hydrogen bonds with other R groups or with a backbone structure. And it's important to differentiate these with the other hydrogen bonds that are in the second structure. These are different because they're only found in the tertiary and quaternary structures. Or these amino acids only make these hydrogen bonds in the tertiary and quaternary structures. And then your last one, or your last type, is going to be nonpolar R groups, and they're going to be making van der Waals interactions. And characteristics of these are just going to be basically hydrophobic. And so if you want to think about the structure function of that, is that if you have a hydrophobic molecule, right, and you place it in a say you place it in a water solution or hydrophilic solution, it's going to want to clump up, right? It's going to want to avoid its solution that it's in. So if you think about in a molecule or a protein that has van der Waals interactions, the hydrophobic effect of it is that those polar groups are going to cluster towards each other on the inside or in the core of the molecule versus um, the exterior, which is going to be composed of the hydrophilic molecules, and vice versa too. Say you have a solution that is, say you have an oil or another like hydrophobic solution, you're going to then have your van der Waals interactions on the outside or the exterior, and then you're going to have your hydrophilic groups form the core, the interior. Is there any like confusions or questions on that? Well, no. All right, so we can go to the next slide. And then we have disulfide bonds. These bonds are going to be your only covalent bonds, and they're only going to happen between cysteine amino acids. And pretty much all you need to know for these is that they stabilize the tertiary and quaternary structures very, very well, and they're the only covalent bonds um, in your tertiary and quaternary structures. All the other ones that we talked about for your electrostatic, ionic, van der Waals, and all that, those are all non-covalent. And the important thing for that is that, remember, covalent bonds are a lot stronger than non-covalent ones. So you're going to find, again, if you think about structure function, you're going to find disulfide bonds in proteins that need to be rigid, that need to be firm, that don't need to be, changing shape that often, that don't need to be that versatile. And another important thing that she mentioned in the videos is that disulfide bonds are good for proteins or molecules that need to be outside of the cell, right? So you think about an antibody protein or molecule. It's going to be in the bloodstream. It's going to be in an extracellular environment, so that's going to be a little bit more harsher. So it's going to need those disulfide bonds to add the support and the structure that it needs, so it doesn't, denature as quickly. But yeah, I think that's all I have on disulfide bonds. So next slide. And yeah, here is just a bond summary. So right, you have two main categories. Either you have covalent or non-covalent. And pretty much for covalent bonds, you're just going to have peptide bonds, which are going to be the bonds between amino acids in the primary structure. And those are only going to be forming your backbone interactions, so they don't really have an effect on the overall shape. And then you're going to have your disulfide bridges or bonds, which are again going to be between your cysteine amino acids, and they're going to be important for molecules that need that extra firmness or rigidity, say if they're in a harsher environment, or say if they don't need to be changing shape that often. And then you have your non-covalent bonds. These are your ionic bonds. Those are between two polar charged amino acids, electrostatic, which is between a polar charged and a polar uncharged. Your hydrogen bonds between the uncharged and your van der Waals interactions. And those are going to be in your tertiary and quaternary structures. And yeah, as far as your secondary structure, those are just going to be the non-covalent hydrogen bonds. And again, those are going to be separate than the amino acid hydrogen bonds. But yeah, that's pretty much it for bond summary. Oh, and again, any questions so far? Unless I forget, no? Okay. It's good to hear. And then here we have another summary of protein structure. All right, I'll just go through this quickly. You have your primary level, and that's just the sequence of amino acids, right? So here we have an example of serine, aspartame, cysteine. And they're going to be stabilized by the peptide bonds between them. Those are going to be covalent bonds, so they're going to be pretty stable relative to the other types of bonds. And then you have your secondary level, and these are just the formation of alpha helices or beta plated sheets in the polypeptide. And they're going to be stabilized by hydrogen bonds between the groups that form the backbone, right? And then you have your tertiary, and that here's where you get your overall three-dimensional shape, right? The protein starts to fold on itself. based on the amino acid interactions, right? The bonds that that forms will determine whatever shape it has, along with the beta plated sheets and alpha helices. Um, yeah, and those bonds are going to be between your R groups, or between the R groups and the backbone. And then you have your final quaternary structure. Not every protein has a quaternary structure, but if it does, it's just going to be a combination of different polypeptides. Um, and again, these are going to be your subunits, not to be confused with your domains. Subunits are just sections of polypeptides or polypeptides themselves. And yeah, they're going to be stabilized by your bonds and other interactions between R groups and between R groups and polypeptide backbones of different polypeptides. So yeah. Any questions there?
No.
Okay, great. And yeah, here we just have an example of two different quaternary structures. You can see on the left one, you have two alpha chains, and then two beta chains, right? So you just have four different polypeptides that come together to form the overall protein. And yeah, the same thing on the right. There's just a different diagram of that. But yeah, that's pretty much it. I don't know if you guys have any questions on anything else, or if Michelle has anything else to add. But
No, I think that's pretty much it for the review. We covered most of the material on Wednesday, so this is just the second half. So yeah, that's all there is. Thank you.
Thank you for setting this up.
yeah, thanks.
I have a
guys for coming.
question. So
Um,
if you have a question about like the problem set during the weekend, can you guys email you?
yeah, of course.
Or no? Okay, perfect. Thank you.
Yeah,
I
yeah.
just
I
didn't
mean,
started yet. I know I should have started yet, but I have other classes. So I
yeah,
was just wondering. Okay, perfect. Thank you so
yeah,
much.
yeah.
Thanks.
You can
Always
email
email us.
TAs anytime.
You can also email in the week too, and we have office hours, or I'm sorry, help sessions every week. The times are on Moodle, so you're always welcome to go to those. So yeah,
Sure, perfect. Thank you guys.
Yeah,
yeah.
thank you.
Saeed, you can stop recording, you know.
Oh yeah.